Hello, dear students. I welcome you all on behalf of Sri Jayendra Rank Zone classes. Dear student, so today we are going to talk about the molecular basis of inheritance. Dear student, in previous chapter of genetics, we have talked about the detailed study about the principle of inheritance and variation. Now, at a molecular level, how this inheritance is getting regulated, that we need to understand. And this chapter mainly focus at a molecular level of inheritance. So in an upcoming lecture, we are going to discuss in detail all about this. But today, in an introductory lecture, we are going to talk about somewhat about the molecular biology and some of the molecules. Molecular biology is relatively young discipline among life science. It is a relatively young discipline among the life science, dear students. Okay, because before some time we people are not interested in knowing the molecular basis or molecular study, but today it is too much research is going on these molecular levels, and it is about the study of macromolecules and their mechanism in living things. We are going to talk about the macromolecule and their mechanism in the living things such as gene replication, mutation, and expression. Okay, so how the gene copying or gene replication occurs, how the mutation occurs, how they are getting expressed into the proteins, gene form a protein, that all things we are going to discuss their mechanism here. It is the field arose from the convergence of the work by geneticist, physicist, and structural chemists. Because geneticist was talking or studying about the genetics, mainly inheritance, and their expression levels. Physicists and structural chemists talk about that detailed study, molecular study, and due to the convergence of all these different thoughts on the structure and function of genes, the molecular biology arose. And in the initial classical period of molecular biology began in 1953. And in 1953, two scientists who actually done a very good work, and from that, the speed of a study in molecular biology is increased. And the true name of the scientist is James Watson and Francis Crick. Great scientists who got a Nobel Prize as well. And their discovery of a double helical structure of DNA. Their discovery of double helical structure of DNA led to the great discoveries further. So, if we talk about James Watson, his full name is James Debe Watson. We call him JD Watson, and who was born in Chicago on 6th April 1928. In 1947, he received BSc degree in zoology, and during these years, his interest in bird watching had matured into a serious desire to learn genetics. In bird watching, he was fond of that, and later on, he particularly seriously took into the what study into the genetics. And this became possible when he received a fellowship for graduate study in zoology at Indiana University, Bloomington, where he received his PhD degree as well in 1915 on study of the effects of hard X-rays on a bacteriophage multiplication. Dear students, please important to note what he in which part he received a PhD. It is the effect of hard X-rays on the bacteriophage multiplication. Later on, he met Crick and discovered their common interest in solving the DNA structure. Both were working on this particular part, and both were very interested to discover the structure of DNA. So their first serious effort was unsatisfactory, but their second effort, based upon the experimental evidences and better appreciation of nucleic acid literature, resulted in the proportion of proposal of complementary double helical configuration in 1953. Complementary double helical configuration in 1953, if we talk, this arise due to the second effect. Okay, Francis Harry Crompton Crick, we call him FHC Crick, who was born in 8 June 1916 at Northampton, England. He studied physics. He was a physics scientist in University College London and obtained BSc in 1937. But their first serious effort was unsatisfactory. He completed PhD in 1954 on the thesis titled X-ray diffraction, polypeptide and protein. A critical influence in Crick's career was his friendship with J.D. Watson, then a young man of 23, leading to 1953 to the proposal of a double helical structure of DNA and replication scheme. So this scientist in introduction or scientist history is of no importance with respect to the exam, but this just for the knowledge, just for the establishing a base that these two different scientists with the common interest met. He received a PhD in different things, but his common interest was to identify the double helical structure of DNA, and he discovered it obviously. So, if we talk about nucleic acid first or DNA, then there are two type of nucleic acid, that's DNA and RNA. DNA we call it deoxyribonucleic acid, and RNA we call ribonucleic acid. These are the two types of nucleic acid found in living system. When we talk about them in detail, dear student, DNA is a D-ribonucleic acid, deoxyribonucleic acid, and act as a genetic material in most of the organisms. We know that. It is acting as a genetic material. It controlling all the activities of a cell. It is present within the nucleus in case of the eukaryotes, and it is present as a nucleoid in case of the prokaryote. And it acts as a genetic material. And if we talk about DNA structure, DNA structure is a double helical model. As you can see here, this is the two chains or two strands of DNA are there. This is the one strand, this is another strand. And these two strands are helically wound around each other. They are helically wound around each other. And in between them, there is a what? A base pairing. So this strand represent a sugar phosphate backbone, okay, and they represent a sugar phosphate backbone, and in between you will find the stack like ladder like structures. These are nothing but a base pair. In base pair you will see the nitrogenous bases like adenine, thymine, guanine and cytosine. As you can see, adenine pairs with the thymine and guanine pair with the cytosine. This is the basic structure. Just I am giving you the hint of what is DNA look like, what how DNA look like, but we will be going to discuss other structure in more detail. So the two strands helically wound around each other, okay, and in between these two strands are joined by the nitrogenous bases like adenine thymine pairing and the guanine and cytosine pairing. Whereas RNA similarly is also a particularly another nucleic acid, and it is a ribonucleic acid which is also acting as a genetic material in some viruses. In viruses we have studied that RNA is the only that it is devoid of protein coat. And this RNA, if we talk, it acts as a messenger. It acts as a messenger and has an additional role as an adapter. We will discuss that RNA are of different type. In this, mRNA will act as a messenger RNA. It passes the information of DNA or information of gene in the DNA to the protein or polypeptide, and that RNA acts as a messenger and has an additional role as an adapter. Okay, it has an additional role as a adapter, means tRNA we are going to talk, which will act as an adapter molecule, and a structural and catalytic molecule. If we talk, we are going to talk about the rRNA. Okay, so these three types of RNA having a different functions. We will discuss that also. So because of this, they act as a messenger as well as they can act as an adapter molecule. They also act as a structural and catalytic molecule. So this is the particularly we can say particular structure of tRNA. It is actually okay, and we will discuss this later on, which is acting as a adapter molecule. Now, if we take a basic questions, then in DNA, double-stranded DNA, just we have seen that adenine pairs with the which one? Thymine. Okay, and Adenine pair with the thymine with the two hydrogen bond. We remember, dear students, if suppose these are the two strands of DNA, these are helically bound. Just I am drawing. These are the sugar phosphate backbone. Sugar phosphate backbone. And here these are the nitrogenous bases. And these nitrogenous bases are joined by hydrogen bond. If it is adenine, then it is paired with the thymine with the two hydrogen. If it is a G, it pair with the three C with the three hydrogen bond. So A pair with T with the two hydrogen bond, and G pairs with C with the three hydrogen bond. Okay, so adenine pair with the thymine. RNA act as a, it is also acting as a particularly a genetic material in some viruses. Okay, it acts as a genetic material in some viruses like HIV, viroids, etc. So another, okay, so these are the two nucleic acids. This, now we are going to discuss about DNA in more detail. DNA is a double helical structural model. It is looking like this. Okay, it is a long DNA, approximately 2.2-meter long DNA, which is packaged into the one nucleus of one cell. If we talk about DNA, it is a deoxyribonucleic acid. It is a helically twisted heteropolymer. Heteropolymer means it is made up of different type of nucleotides, and heteropolymer of deoxyribonucleotides. DNA or deoxyribose nucleic acid is helically twisted heteropolymer of deoxyribonucleic acid nucleotide. And DNA is the largest biomacromolecule. DNA is the largest biomacromolecule. Okay. Now, DNA, if we talk, it represents a genetic material of organism and forms the molecular base of heredity. Actually, the inheritance of character occur from the parent to the offspring, but through which material? So that occur through the DNA, because DNA have a genes and genes have a coding information. So this is a helical structure DNA of particularly we can say a DNA. In this, if we talk, you can see this is the particularly sugar phosphate backbone, and in between you will see this are the base pair. These are the base pair. Okay, so this particularly thing is found here. Now, if we talk about this, okay, then here two sugar phosphate backbone, two strands are helically wound around each other, and base pair is present in between them. Now, here the length of DNA is usually defined in terms of a number of nucleotides or nucleotide pairs, or a nucleotide pair is also known as base pair. Base pair means A, A, T, G, C are separate nucleotides and nitrogen is bases. Okay, and this four different types of nucleotides are found. But if A pair with the T, it is a base pair, and G pair with the C, it is a base pair. So it can be calculated, it can be measured in a number of base pair or number of nucleotide. Bacteriophage known has, if we talk about the length of DNA in a bacteriophage, which is known as 5174, has a 5386 nucleotide. Okay, they have a 5080 5386 nucleotide. Whereas if we talk about a bacteriophage lambda phase, then lambda phase has a 48502 base pair. 48502 base pair. Okay, and Escherichia coli has a 4.6 into 10 raised to 6 base pair. Whereas if we talk about a haploid content of human DNA, then it has a 3.3 into 10 raised to 9 base pair. Base pair like ATGC, AT and GC. These are two different types of base pair are found. How many are there in haploid content of DNA? 3.3 into 10 raised to 9. If it is a diploid content, it is going to be 6.6 into 10 raised to 9 base pair. So this long DNA is seen. Now, if we talk about the DNA is known as a deoxyribonucleic acid. So DNA is nothing but it is a polynucleotide, means it is a chain of nucleotide. It is a structure of polynucleotide or chain, or it is a chain of polynucleotide. And if a chain of polynucleotide, that is a DNA or RNA, then this DNA or RNA made up of so many nucleotides. If we talk about nucleotides, then this nucleotide combinedly form this nucleic acid or this polynucleotide chain in case of the RNA and DNA. Nucleotides are complex biomolecule formed by the condensation of, they are formed by a condensation of pentose sugar, one cyclic nitrogenous base and phosphoric acid. Dear students, please remember one of the important point that here you need to remember, that nucleotide are made up of pentose sugar, one cyclic nitrogenous base and one phosphoric acid. So here you can see in this diagram, this is a one particularly nucleotide. Here it is a pentose sugar, okay. Here it is a phosphate and here it is a nitrogenous base. This combinedly form a what particularly nucleotide. But if we talk a nucleotide, then nucleotide consists of two things. That is nucleoside and phosphoric acid. Now consists of nucleoside consists of nitrogenous base and pentose sugar, okay. So this way nucleoside and phosphoric acid is nucleotide, whereas nitrogenous base and pentose sugar is nucleoside. Now nitrogenous bases are of different type that we will discuss. So nucleotides like ATP and GTP function as a energy transfer system of the cell, and ATP is considered as energy currency of the cell. So ATP and GTP are what? Nothing but they are the nucleotide. Why? Because in ATP the nitrogenous base is adenine, and in GTP nitrogenous base is guanine. So nucleotides of nucleic acid function as an information transfer system of the cell. Nucleotides nucleotides of a nucleic acid function as information transfer system of the cell. Okay, they are able to transfer information particularly of the cell, because this nucleotide are present in the DNA, and DNA has a particularly the code of this nucleotide. That code has the information of any protein or any, and which gives a particular character to the particular cell. Okay, so in this there are three components in a nucleotide, and nucleotide combinedly going to form our DNA or RNA. Nucleotide going to have a pentose sugar, nitrogenous base, and the phosphoric acid. First, we are going to talk about the pentose sugar. Dear student, pentose sugar, if we talk, it is present in nucleotides are of two types. There are two types of pentose sugar are there. In DNA, you will see that deoxyribose sugar is found, whereas in RNA, which is also nucleic acid, there ribose sugar is found. If we talk about the chemical formula of a deoxyribose sugar, then it has a 5 carbon, 10 hydrogen, and 4 oxygen, means C5H10O4. Whereas ribose sugar have a C5H10O5. Okay, so here one oxygen is more in case of the ribose sugar, but because and one oxygen is less in case of the deoxyribose. So ribose sugar has a total 5 oxygen, but when one oxygen lack, then it is going to convert into the deoxyribose sugar. So because of this, there is a lack of or absence of one oxygen, or less one oxygen is there in deoxyribose sugar. Now both the pentose sugar, if we talk, they occur in a pentagon or a furanose ring, or 5 carbon ring we can say, which contain 4 carbon, sorry, it is a pentagon shape, and Furanose ring having a means four carbon and one oxygen ring. The fifth carbon lies outside the ring. So you can see this is the structure of ribose and deoxyribose sugar. Here in deoxyribose sugar, this is the oxygen. This is the pentagon shape. They have four carbon. On the first carbon, you will see the presence of OH and H. On the second carbon, you will see the H and OH. Okay. And third carbon also you will see the H, OH. Okay. And fourth carbon you will see the H, and on fifth carbon you will see that that is OH and H2. Okay, so CHO. Okay, this is found here. So CH2OH is going to be formed here. So this is OH is linked with this, and there is a CH2. So here also you will see when deoxyribose is there, the substance structure is similar to the ribose. Just on the second carbon you will lack the particularly one oxygen compared to here. Oxygen compared to here. So because of this, it is known as a deoxyribose. Here in this fifth carbon lies on the outside, the ring. Dear student, this furanose ring having a structure and two ribose and deoxyribose sugar are found. In deoxyribose, you will see hydroxyl groups are formed only on the carbon atom 1, 3, and 5. See, 1, 3, and 5. In case of the deoxyribose, actually 1, 3, and 5. Here also you will see the OH group. But, and you can see here also in this, here also it is shown. In deoxyribose, second carbon atom is replaced by hydrogen. So here OH group is replaced by hydrogen. So here in this, if it is a deoxyribose, then it is a CH2OH, H, like this 5 carbon are there. This 135 have a OH group. And here, so the second graph, in ribose sugar, you will see presence of OH, and here you will see presence of H. In a ribose sugar, the hydroxyl group are found only on carbon atom. That is first, second, third, and fourth. Whereas in deoxyribose, at the second carbon, you will not see the presence of hydroxyl group. You will see the H group. So in ribose sugar, you will see the one hydroxyl group on the first, third, fifth, first, second, third, and fifth carbon. Then there is a nitrogen base. So these two different type of sugars are found. Ribose sugar is found in RNA, and deoxyribose sugar is found in DNA. Now, apart from this, in nucleoside, you will see the presence of nitrogenous base. Now, nitrogenous phase, if we talk, they are nitrogen containing heterocyclic compound. Why they are known as nitrogenous bases? Because they have a nitrogen in their cyclic compound, and they are of two types, purine and pyrimidine. If we talk about purine, they are a 9-membered dicyclic or double ring nitrogenous bases. They are particularly 9-membered or dicyclic or double ring nitrogenous bases, where the two rings are joined at fourth and fifth position. And nitrogen occur on the 1, 3, 5, 7, and 9 position. So if we draw the structure of this purine, it is like this. So this type of ring is formed. See, this is the first carbon, this is second, this is third, this is fourth, this is fifth, this is sixth, this is seventh, eighth, and ninth position. So on the first, third, seventh, and ninth position, you will see the presence of nitrogen. You will see the presence of nitrogen. Okay. Now, here, dear student, if we talk, these two rings are there. This 6-membered ring, and this is a 5-membered ring. Okay. Okay. So these two ring combinedly form uh, purine. And when we talk about this purine, these purines are of two types. That is the adenine and the guanine. Adenine is represented by A and guanine is represented by G. Now both of them occur in nucleotide of DNA and RNA. Adenine and guanine both are found in DNA as well as RNA, and the structure are like this. So here, if we talk on this particular adenine, this is the first position, second, third, fourth, fifth, sixth, seven, eight, nine. Now here on the fourth carbon, you will see the NH2 group. Okay, and here this particular thing is found. So in this adenine structure, you will see that at fourth carbon, you will see the presence of amino group. Okay, so actually this carbon numbering is not like this. This carbon numbering is like this. Okay, that we need to note down here. This carbon numbering is like this. That is the first carbon. This is second. This is third. This is fourth. This is fifth, and this is sixth. Okay, then this is seven, eight, and nine. So here at the sixth carbon, you will see that there is a at the sixth carbon, you will see that there is a presence of amino group. So we can call adenine as a chemical name, that is a 6 amino purine. 6 amino purine. When we talk about guanine, in guanine also, if you remember this like this, 1, 2, 3, 4, 5, 6, 7, 8, 9, 9 number ring. Here on this second carbon, you will see the amino group, and on the sixth carbon, you will see the particular that is what oxy group. Okay, so this guanine is named technically as 2 amino 6 oxy purine. It is a 2 amino 6 oxy purine. So this 2 amino 6 oxy purine is guanine, whereas 6 amino purine is nothing but adenine. And this adenine and guanine both are the purine nitrogenous bases. Whereas if we talk about the pyrimidine nitrogenous bases, they are 6 membered monocyclic ring or single ring nitrogenous bases. They have a nitrogen as first and third position, and they are of three type, that is cytosine, then thymine, and uracil. If we talk about the structure, the pyrimidine ring is like this. Okay, so this way the particularly the things happen. Okay, the nitrogen are at first and third position. Okay, now like this we can stabilize them. Okay, so this is of three type, that is a cytosine, thymine, and which are the pyrimidine. Now here this cytosine is found in both DNA and RNA. So this is found in both DNA and RNA, that is cytosine. If we talk about thymine, then this thymine is particularly, we can say it is found in DNA, whereas uracil is found in RNA. Now this pyrimidines, if we talk, In this, cytosine is common to both nucleotide of DNA and RNA, and the structure of cytosine is like this. Okay. Now, if you see the structure of the cytosine, you will see that on this fourth carbon, you will see the presence of what? Particularly, cytosine is chemically known as 2-oxy 4-amino pyrimidine. If it is known as 2-oxy 4-amino pyrimidine, then it is the second carbon, this is the fourth carbon, then it is going to be third carbon, this is the first carbon, this is fourth carbon, and this fifth carbon. Okay, so like this, the particular structure is there. So the 2-oxy 4-amino purine is a cytosine, whereas thymine is found in deoxyribonucleotides. Okay, now in this structure, we remember that here on the first carbon, first position, this is nitrogen, and this nitrogen is attached with the hydrogen. Okay, on the second, it is a carbon. Okay, and this carbon is attached with the oxygen. Then this is nitrogen, this is carbon, and here this is a nitrogen, this is hydrogen, this is hydrogen. Whereas here on the fourth position, sorry, this is the fifth position, this is sixth position. On this fifth position, you will see this is a carbon and this is a hydrogen, and on sixth you will see the carbon and hydrogen. So if we make its structure in a proper way, then nitrogen, here it is hydrogen, you can make like this also, C double bond O and double bond C, CH, CH, and N. Here CNH2, C double bond O, and this way, if we number 2, 3, 4, 5, 6 at 1 and third position, you will see the nitrogen, whereas second, fourth, and 5 and 6th position, you will see the carbon. Now, when we talk about the thymine, it is found only in deoxyribonucleotides or in DNA, and the structure is like this. In this, if we talk about the structure of this thymine, it is 2,4 dioxy 5 methyl pyrimidine. Now 2,4 dioxy, if we talk, then it is a second carbon, this is fourth, this is fifth, this is one, this is third, and this is sixth. On the 2 and 4, you will see the presence of oxygen, but first and third position have a nitrogen, and fifth position you will see methyl CH3. Okay, so this way whole structure forms. So if we make its particular structure like this, then the thymine look like somewhat like this. If it is M, then match with the H, carbon, nitrogen, carbon, 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 carbon. Now at second, first, second, third, fourth, fifth, and sixth. At second carbon, you will see the oxy group. At the fourth carbon, also you will see the oxy group. So here we put N, so three valencies substituted, and the carbon have a CS3 group at the fifth carbon. So like this, the valency is completed. The structure is 2,4-dioxy 5-methyl pyrimidine. So that is a thymine which is found in DNA. Now the another nitrogenous base, that is uracil, occur in only ribonucleotides, or we can say only in RNA. And 5-methyl uracil is the other name for the thymine. Okay, thymine is also named as what? 5-methyl uracil. So we can write here also. So 5-methyl uracil, at fifth, if you add a methyl group, then it is going to be it is modified into thymine from the uracil. So in uracil, You will not see this methyl group. Apart from this oxy group you will see. So if we talk about the uracil name is going to be 2,4-dioxypyrimidine. 2,4-dioxypyrimidine. And in 2,4-dioxypyrimidine, if we talk, you can see this is the nitrogen, carbon, nitrogen, carbon, carbon, carbon. And here at the second group, you can see oxy group, and fourth group, you will see oxy group. If we add a fifth group, it methyl group, then it is going to be converted into the thymine. But this is the uracil, and uracil is found in case of the RNA only, and it is a pyrimidine. Okay, dear students, so this is all about the nitrogenous bases. Now, nitrogenous bases present in a form of base pair in DNA and RNA. And DNA RNA somewhat look like this. If this is the DNA, you can see this the two strands are helically bound around each other, and in between you will see this particular base pair. And in this base pair, you will see the cytosine, guanine, adenine, and thymine bases, where adenine pairs with thymine and guanine pairs with cytosine by 3 hydrogen bond. Whereas in RNA, you will see which is usually single stranded, and here it has a nitrogenous basis. Okay, and this is a particularly cytosine. Okay, guanine, adenine, uracil. In case of uracil is present in case of thymine. Thymine is found in case of the DNA, but uracil is found in case of the particularly we can say RNA. In RNA, if guanine pairs with C, then obvious if pairing is found in a double strand RNA, then A is going to pair with the U. Okay, this is the thing. Now, dear student, apart from the nitrogenous base in pentose sugar, we have discussed the phosphoric acid is found in the polynucleotide chain or a DNA, and it is chemically S3PO4 or PO4 3 minus, and it provides a final covalent bond that connects sugar in a series. So in a strand, if you want to connect a sugar, because nitrogenous bases are protruded inward, if it is a DNA strand, if it is a DNA strand, the nitrogenous bases are protruded like this inward, but here this sugar are there. Now this sugar linked to each other in a series by this covalent bond by the use of phosphoric acid. So the backbone of nucleic acid stand in a chain of alternating phosphate sugar molecule. So if it is a sugar, here you will say find a phosphate, then you will see sugar, you will find phosphate, then you will see sugar. So it is known as sugar phosphate backbone. Dear student, in phosphoric acid there are 3 hydroxyl group and 1 oxygen attached to the central phosphorus. So you can see the structure here, that phosphoruses are there and 3 hydroxyl group are there and 1 oxygen which is with the double bond. Association of phosphoric acid in nucleotide also provide acidic trait if it is a phosphoric acid. So if we add it into the nucleotide or into the nitrogenous bases or and sugar, when it nucleotide form, it gives acidic nature to the nucleotide. Because of this, when we talk about DNA or particular deoxyribonucleotide gene, that is a DNA, we call it as a what, deoxyribonucleic acid. Same way in RNA, the ribonucleotide chain, we call it as a ribonucleic acid due to the presence of phosphoric acid. So nucleoside, if we talk, it is a condensation of product of pentose sugar and nitrogenous base. So we have understood this, that nitrogenous base and pentose sugar combinedly form a nucleoside. And the linkage established between the pentose sugar and nitrogenous base is called a glycosidic linkage. We remember, glycosidic and a glycosidic linkage is formed between the nitrogenous base and sugar. So in purine nucleosides, glycosidic bond is established between the first carbon atom of pentose sugar with the ninth nitrogen of the C. This is the particularly we can say two amino purine. Suppose adenine, it is adenine. Okay, and in adenine, if we talk, this is the ninth nitrogen, 7, 8, ninth nitrogen, and this is a pentose sugar. In this is the first carbon. Now carbon of sugar are represented by a dash. So this is a glycosidic linkage, but this glycosidic linkage established between first carbon and the ninth nitrogen of the purine. Okay, so this way the particularly we can say occurs. But in guanosine or in purine, if we talk about the guanine and the sugar, then also it is between the ninth nitrogen and first carbon. So here also in guanosine also 1 dash 9 we can say the glycosidic bond is established. In pyrimidines nucleosides, if we talk, which are formed like this, see this is the nucleoside because phosphoric acid is not there. So in pyrimidines nucleoside, the glycosidic bond is established between the first carbon atom of pentose sugar with the first nitrogen atom of the pyrimidine. Depending upon the pentose sugar involved, nucleotides are of nucleosides are of two types, that is deoxyribonucleotides and ribonucleotides. Here you can see the cytidine and uridine. So this is of the particularly RNA. And in RNA, if you talk, this is about the cytosine, and this is what a sugar. Here you can see this is the first, we can say nitrogen, and this is the third nitrogen, and this is the first carbon. So 1 dash 1 nitrogen glycosidic bond is established. And same here, if it is a uracil and it is a sugar, then here also this is the glycosidic bond is established, which is the first sugar, first carbon of sugar, and first nitrogen of uracil. Okay, and this 1 dash 1 glycosidic bond is established. So ribonucleosides and deoxyribonucleosides are formed. So uracil forms a nucleoside with the only ribosugar, while thymine forms nucleoside with the deoxyribosugar only, because the uracil is found in RNA and thymine is found in DNA only. So adenine, guanine, cytosine produce nucleosides with both ribose and deoxyribose sugar. So uracil forms a nucleoside with only ribose sugar, while thymine form a nucleoside with the deoxyribose. Adenine, guanine, cytosine produce a nucleoside with the ribose and deoxyribose both sugar. Why? Because uracil is found in only RNA and thymine is found in only DNA. Now, when the nucleoside are formed, they will in DNA, this nucleoside will be named as a deoxyadenosine, deoxyguanosine, deoxycytidine, deoxythymidine. So here what? Deoxyribose sugar plus adenine, deoxyribose plus guanine will form a what? A deoxyguanosine. Deoxyribose sugar plus cytosine will form a deoxycytidine, and deoxyribose sugar plus thymine is going to form a deoxythymidine in case of DNA. But in RNA, in a ribose sugar is going to be a, so we can't use a term deoxy. So the nucleoside DNA nucleoside are named like this, but in RNA nucleoside are named like this, that adenosine, that is adenine plus ribose sugar, guanosine, that is guanine plus ribose sugar, cytidine, that is a cytosine plus ribose sugar, and uridine, thymine is not here, uridine, so uracil plus we can say ribose sugar. So if we talk about some of the MCQs, energy currency of cell, that we know very well, that is adenosine triphosphate, which is a nucleotide. Chemical formula of deoxyribose sugar is deoxyribose sugar. So one oxygen is less, so it is going to be C5H10O4. Sugar in RNA is obviously it is a ribose sugar. Okay, dear students. So I hope you understood this particularly nucleotide and nucleoside difference. Now, after studying this particular nucleoside and nucleotide difference, and in RNA and DNA, what type of nucleotides are found, how this polynucleotide chain are formed, we have to understand the structural model of DNA, which is given by Watson and Crick, and that we will continue in the next class. Thank you.